Bon, ben, bonjour tout le monde. Merci d'être là. Je suis vraiment contente de. Tout le monde m'entend, oui. Je suis vraiment contente qu'il y ait autant de gens qui soient présents pour la présentation d'aujourd'hui de Daniel Bolnick. Daniel est professeur à l'University of Connecticut dans le département de Ecology and Evolutionary Biology. Daniel a fait son doctorat à l'University of California Davis avec Peter Wainwright et son postdoctorat aussi à UC Davis avec Michael Turelli. Ensuite, tout après, il est devenu professeur à l'University of Texas à Austin en 2004. Et dans cette période-là où il était au Texas, il était aussi en même temps Early Career Scientist au Howard Hughes Medical Institute. En 2018, il a déménagé son laboratoire à l'University of Connecticut, donc plus proche de nous. On est chanceux pour ça. Et d'ailleurs, il pensait venir en voiture nous faire sa présentation ce printemps, mais ça ne s'est pas réalisé pour le moment, en tout cas. Daniel, c'est l'éditeur en chef de la revue American Naturalist. Donc, si vous voulez lui poser des questions là-dessus aussi, sur toute la partie publication de vos recherches, c'est aussi une bonne personne à qui on peut poser des questions. Je ne vous listerai pas ces 150 publications scientifiques, là, mais je suis sûre qu'après sa présentation, vous allez avoir envie d'aller les lire. Son laboratoire, on peut dire que c'est un laboratoire d'éco-évo-immunodynamique. C'est un précurseur dans le domaine qui va marier l'évolution, l'écologie, l'étude de l'immunologie. Il travaille sur différents sujets en évolution depuis que son laboratoire est ouvert, incluant les adaptations à échelle géographique restreinte, la saltative mating, la divergence en présence de flux géniques, les variables environnementales et génétiques qui affectent le microbiote des individus. Je pense avant que ce soit même un concept dont on parlait dans les journaux là, que les gens connaissaient, lui étudiait déjà ces questions-là. Et leur espèce de prédilection, c'est l'épinoche à 3 épines. Un poisson qui est vraiment beaucoup étudié en écologie, en évolution, aussi en biologie comportementale et en génomique. D'ailleurs, plusieurs laboratoires travaillent sur cette espèce à l'Université Laval, à l'IBIS. Aujourd'hui, il va nous présenter un volet de sa recherche en immunologie évolutive, un sujet dont il est un des chercheurs pionniers. Il va nous présenter ses travaux, les travaux de son groupe sur la coévolution entre un hôte et son parasite en utilisant le système fascinant de l'épinoche à 3 épines et Schistosephalus solidus, que vous connaissez déjà. Si vous avez suivi les travaux de mon laboratoire et du labo de Christian Landry sur les parasites qui manipulent potentiellement le comportement de leur hôte. Nous, on travaille sur la partie génomique et comportementale et le labo de Dan fait tous des aspects vraiment intéressants de l'évolution de la résistance et de la virulence. Thank you, Daniel, for taking the time to talk to us in this very stressful week in your country. And now I'll let you take the stage.
Okay, well, thank you very much. I'm afraid my French is not worth parading in front of you. I speak a little bit. I was able to understand most of what Nadia said, but I wouldn't be able to complete a full sentence anymore. It turned into Swahili halfway through, and that wouldn't help any of us. I'm very pleased to be able to talk to you, although it would have been very nice to visit in person. There are a few familiar faces in the audience, and people would be very much fun to sit down and have a long conversation with. And also, I've been looking forward to spending more time in Quebec. My mother's side of the family are Quebecois. Surnames like Ouellette and Fonjemi and Valdebancourt pepper my family tree, and it would be very fun to spend more time visiting the areas where half of my family comes from historically. Another time, I'll make it up. It's actually not so far a drive. It's a reasonable long day drive. So I'm looking forward to talking to you today. And what I'll tell you about is material that is the culmination of about a decade's worth of work. And some of the results I'll tell you about are 10 years old, and some of the results I'll tell you about are less than a week old, maybe 2 weeks old now. And together it paints a picture of what we're referring to as an evolutionary pyrrhic victory. And because I don't want to assume that the language translates entirely, let me just explain what a pyrrhic victory is. So this is a reference to a Macedonian general, King Pyrrhus of Epirus. And King Pyrrhus invaded the Italian peninsula in about 290 BC. And he won every single battle that he fought against the Romans, in part because he was the very first person to bring war elephants into play against the Romans. And the Romans had no idea what to do against war elephants. This was long before Hannibal. And so he won every single victory that he fought, but he had no mechanism for replacing the soldiers who he lost in battle. And as a result, his army got smaller and smaller with every successive victory. And he is allegedly he said, if we are victorious in one more battle with the Romans, we shall be utterly ruined. And so despite winning every battle, he had to retreat back to his home and leave most of the winnings that they had taken from Roman towns over his conquest. And effectively, he was defeated, even though he never lost a single battle. So today we use the phrase pyrrhic victory to represent a situation where somebody wins, but they wins in such a damaging way that it's maybe not worthwhile doing. And so I'm going to tell you about an evolutionary pyrrhic victory in which my favorite study organism, the three-spined stickleback, in some populations has won the battle against a certain parasite, but maybe in a way that is not worthwhile. But before I launch into that, let me start with another quote. Sorry. There we go. I'm actually going to do a quick adjustment here for my screen share because it's not going to toggle between things the way I would have liked. So give me just a second. Switch to presenter view and change your view here. Okay, sorry about that. Okay, so another quote that I really like, aside from the pyrrhic victory one, is this quote from Bernd Heinrich in his book, Ravens in Winter. And let me back up a second. So Bernd Heinrich, in his book, discusses the, I'm having difficulty navigating my, okay, there we go. Okay, problem solved. So in his book, Ravens in Winter, Bernd Heinrich talks about becoming interested in the behavioral ecology of ravens in the boreal forest in Maine, and in particular puzzled by a behavior that he thought very odd, which was ravens were sharing food with each other in winter. They would find a carcass, a dead deer or a moose, and rather than keeping it to themselves, even though it will remain frozen and edible for a very long time, they would go and get other ravens to share it with. And his book is about the process of exploration and the series of experiments that he tried and failed and tried other things to understand why ravens would do this puzzling thing. And he opens his book by commenting on the fact that this sharing behavior is something he had noticed for many years, but never really paid any attention to. And I think this is generally true. And so I really love this phrase. It's amazing how you can see something every day and yet not notice it. And I think often when we do science with a particular hypothesis in mind, and we develop blinders that focus our attention on just the hypothesis that we're seeking to answer. And in the process of doing that, we maybe lose sight of some things that are actually very interesting along the way that we see but don't pay close attention to. And that's certainly true for me. I spent many years working on the evolution of feeding behavior in three-spined stickleback and feeding morphology, and completely ignoring something that I saw every day, but I didn't pay close attention to. And that was this tapeworm, Schistocephalus solidus, that I suspect many of you are already familiar with from Nadia's own research. So this tapeworm lives inside the body cavity of stickleback. And yes, that tapeworm did in fact come out of that individual fish. And so I spent many years seeing this, but not asking about it. And I would also see something that perhaps you might have missed in this very first slide, and I didn't pay close attention to either for a very long time, which is the fact that there are sometimes dramatic differences in the sizes of parasites. So there's another tapeworm hiding right there as well, inside that circle. That variation in tapeworm size can easily be dismissed as a consequence of maybe one is younger than the other, a more recent infection. But it turns out that when we started looking more closely, some of this variation in tapeworm size was itself biologically interesting and not just noise. And if that's not enough to turn you off of sushi, here's another infected stickleback. And again, all of those individuals came out of that particular fish. So I did in fact see this tapeworm in my very first field season and laboratory work after that. And so I've been seeing Schistocephalus solidus for about 20 years at this point. And yet it took me almost a decade before I started paying close attention to it. So I was working on stickleback to understand how resource competition for food drives disruptive selection that can favor the maintenance of genetic variation inside populations. And so we were dissecting fish to figure out what they were eating and to sex them and to get measures of their investment in reproduction and in growth. And so we would see the parasite and not really do anything with that information except record it as a covariate when we're interested in which individuals are reproducing and which are not. And it took a lab technician, Anli Lau, who was dissecting about 5,000 or 6,000 stickleback for me for a project spanning many, many lakes on Vancouver Island. And she systematically recorded every one of the tapeworms that she saw. And we ended up with a very large document with data on which lakes have which parasites and how severe they are. And again, that was just as a covariate in our statistics, not something we were asking about specifically. And it took a graduate student in my lab, Will Stutz, to open up that data file and treat the parasites as the main story instead of a covariate. And so Will became interested in documenting how the prevalence of this tapeworm varied from one location to the next, leading to the following figure. Every dot here is a different lake on Vancouver Island. And what you can see clearly is that some lakes have tapeworm prevalences on the order of 70 or 80% of fish being infected. Other lakes, maybe it's 5 or 10%. And there are a fair number of lakes where we have never seen the tapeworm. Some of them, like Roberts Lake, we have at this point over 2,000 fish sampled over the past 20 years, and we've never seen a single Schistocephalus solidus from that lake in 20 years. And so Will both documented this variation and began the process of asking, why is it so variable from place to place? To what extent is this variation a function of the ecology of the lakes? To what extent is it a function of the morphology and ecology of the fish? And to what extent is it a function of the immunity of the fish? And so that will be the first topic in today's talk. Should we be ecologists or should we be immunologists? Then part of the answer, of course, will be we should be immunologists at least some of the time. And so I'll then tell you a little bit about both how effective that immunity is and some of the costs. And that's where we'll get at the pyrrhic victory side of the story. And then we'll end up delving into what's really the main obsession of my lab right now, which is understanding the genetics of this immune response. So to introduce the system first, before we dive into our 3 acts of the play today, the main character here is the 3-spined stickleback, which is a very charismatic animal. Here's a female ignoring a male, even though he's nice and brightly colored. And he looks a little distraught. One of the reasons they're such a useful organism is, as you can see in the background, there are a large number of individuals just swimming around all the time. So they are very abundant. They're very easy to capture. They're very easy to study in the wild. And it's a beautiful place to work. There are many lakes on Vancouver Island with spectacular variation, ranging from very small lakes to very big lakes. And that ecological variability between populations makes it a very attractive place to do field work. The other main feature that makes Vancouver Island such a great place to do these studies is that 12,000 years ago, 15,000 years ago, there were no lakes or streams to be found. The island was under a kilometer or 2 kilometers of ice. And as the Pleistocene glaciers melted, oceanic stickleback colonized freshwater populations and repeatedly and independently established replicate cases of adaptation to fresh water. And as they do so, these different watersheds represent parallel evolution of adaptation to, say, large lakes, but there's also a great deal of variation in lake size and the kinds of habitats in fresh water. And so we see very different phenotypes from one population to the other. These are all freshwater lake stickleback. These are all breeding males taken from nests. So they are all successfully reproducing, and they are from different lakes. And so there are lakes with red males or blue males or black males. There are lakes where the males are indistinguishable from females. So there's so much phenotypic variability here. And of course, the most dramatic phenotypic variation that we know of is between the benthic and limnetic species pairs that inhabit 6 lakes in British Columbia. And I want to emphasize this, the vast majority of lakes are not these benthic limnetic species. But these species are fantastic because they are reproductively mostly isolated, they're ecologically different, and they eat different prey items, and we'll come back to this in a minute. The limnetic species eats midwater zooplankton, and the benthic species eats large macroinvertebrates. And their morphology differs as a result. So here's a micro CT scan of the stickleback skull. And what I want to emphasize here, if I back up a second and play this video again, having screen share difficulties. Okay, here we go. Now it should play. We're going to turn the skull and we're going to zoom in inside the mouth of the stickleback here. And you'll see these comb-like structures on the gills. Right there, you see those combs? Those are the gill rakers that are used to filter food items from the water column. And benthic species have very short gill rakers, limnetic species have very long gill rakers, and it's very discrete. It's quite clear which is which. But in the vast majority of lakes, we have a single population per lake that's morphologically intermediate. They're normally distributed, and the individuals at the tails of the distribution here are more benthic or more limnetic. So this population is not two discrete morphs, it's a continuous phenotype distribution. And yet, we see that individuals in one group, so this is a scatter plot using stable isotopes to measure diet in one lake, and superimposed on here are the averages for limnetic species in blue and benthic species in green, paired up. And you can see the individuals in this one lake, the dots, span more ecological variation in diet than the benthic limnetic species do. So inside a single lake, you see more ecological variability than between these species pairs. And that's important because, well, we'll get to that in a second. The last thing that I want to say is that although we see this full range from limnetic individuals to benthic individuals and generalists in between, the average phenotype varies from lake to lake. Very small lakes are mostly benthic prey availability, and so we have mostly benthic foragers. In very big lakes, we tend to find, on average, limnetic feeding populations. So inside every one of these lakes, we see variation, some benthic and some limnetic individuals, but you range from being a population that is mostly benthic to populations that are generalist intermediates to populations that on average are limnetic specialists, although you always have variation inside the lakes. But this is important because one of the food items in question here is this copepod, and limnetic fish tend to be more likely to eat copepods, and those copepods are the intermediate host, well, the first host, I should say, for this parasite. So the life cycle for Schistocephalus solidus, the tapeworm eggs are created inside bird intestines where the tapeworms mate. The eggs are defecated, hopefully into water, where they hatch, and they enter copepods, which are eaten by stickleback. And only 3-spine stickleback are suitable hosts for this particular parasite. Then any of a number of piscivorous birds can eat an infected stickleback, and the tapeworm mates inside the bird. the tapeworm grows to its full reproductive size inside the stickleback. And it can do that because it doesn't exist in the intestines. Instead, when the fish eats the copepod, the tapeworm burrows through the intestinal wall into the body cavity, and it grows inside the body cavity among the organs. So with that for background to introduce our main characters, how much of the variation in this tapeworm abundance between lakes is due to ecology, given that you get the tapeworm by eating an infected copepod? So maybe it's just the difference between benthic and limnetic feeding populations. So again, here's this variation that we're trying to explain. Is that differences in exposure risk? The populations on the right maybe are more limnetic, they eat a lot of copepods, and the left are more benthic. Or maybe that's not the story, and instead we have susceptible populations on the right and resistant populations on the left. Which is it? Well, to begin with, ecology certainly matters. The anadromous stickleback, at least on Vancouver Island, tend not to get schistocephalus because the tapeworm does not hatch well in brackish water. And so the stickleback that are marine, who come in to breed only for a short time in estuaries, are mostly eating other copepods, and they're not infected, and so the fish themselves are not exposed. Stream fish don't eat as many copepods either, so they don't have as high an abundance of the tapeworm, but lake fish frequently, though not always, are eating plankton. So the marine fish we've confirmed through infection experiments tend to be susceptible to infection, whereas lake populations tend to be resistant. So then why is there so much variation still among lakes? Well, within lakes, remember we have limnetic and benthic individuals, and we can use that variation inside lakes to connect and predict tapeworm infection status. So individuals who morphologically are predisposed to eat limnetic prey, these are smaller fish with longer gill rakers, are statistically more likely to be infected with the cestode than fish that look more benthic-like morphologically. So at the individual fish level inside lakes, the more limnetic an individual is, the more likely it is to have this parasite that is acquired by eating limnetic food. The same, it turns out, holds across lakes. The first study we did, we found a non-significant trend. We have since added more data. And so we find that populations with, on average, longer gill rakers, a very limnetic trait, are more likely to have high-intensity schistocephalus infections. So again, the ecology makes sense. The more plankton an individual eats, the more plankton a population eats, the more we see Schistocephalus solidus. And yet there's still a lot of residual variation. How much of that residual variation maybe is immunological instead of ecological? And to address this, we're going to look at two populations highlighted here that differ substantially in their infection rate, but not in their diet. So this is Roberts Lake and Gosling Lake on Vancouver Island. In both lakes, cyclopoid copepods are 5% of prey items in the stomachs of the fish. Loons, a common terminal host, nest in both lakes. So ecologically, everything is quite similar for infection risk. And yet, over the span of nearly 20 years now, I should say 15 years, there's persistent differences. Gosling Lake fish are always heavily infected. Roberts Lake fish, we still have never confirmed a tapeworm in any Roberts Lake fish from the wild. And I'm going to use these icons here to distinguish the fish in the future. The infected photograph here I'll use to remind you that Gosling Lake is heavily infected. And I'll use the happy, bright red, blue-eyed male on the bottom to remind you that Roberts Lake are the rarely, if ever, infected population in the wild. So to figure out if these are immunological differences instead of ecological, we did what comes naturally to a geneticist. And this is work with postdoc Jesse Weber and Natalie Steinel and my graduate students, Brian Lohman and Will Shim. We experimentally raised fish from these two populations, pure crosses and F1 hybrids, and we experimentally exposed them to either copepods with the tapeworm or copepods without the tapeworm as a control. And what we found is that the rarely infected population has a stronger granulocyte induction response. After they're infected, we see a big proliferation of granulocytes measured via flow cytometry. Those granulocytes generate more reactive oxygen. So these Roberts Lake fish have both more cells and more aggressive cells in this immune response. And there's a weak tendency for them to have a lower infection rate in lab experiments. It was not significant in the first experiment we did. In a follow-up experiment, it was statistically significant. Combining the data sets, it's an overall significant effect, but it's weak. So the Roberts Lake fish do get infected in the laboratory, just not quite as much. But where we really see a big difference is in the size of the tapeworms. The tapeworms shown here are both 43 days old. They are all full siblings to each other because we bred them in the laboratory. The difference is the genotype of the fish that these two groups of tapeworms came out of. So to show this to you statistically now, here's the data. We have the highly infected population on the top and the rarely infected population, if ever, on the bottom. Those are the pure lab raised types. F1s and F2s are in the middle. Back crosses are on either side. And this is log cestode mass. And so you can see very, very strong host genetic control of tapeworm mass. And this is about 2 orders of magnitude variation in tapeworm mass. So a huge effect on the growth rate of the tapeworm, which matters because if the tapeworm does not get big enough, then it's not able to reproduce when it gets into a bird. So to answer the questions from Act 1, the answer, of course, is both. We need to be ecologists and immunologists because a fish's limnetic diet enhances its exposure risk. But some populations can minimize that by being immune, and they reduce cestode establishment and growth. Other populations do not. And so the next question is, why do some populations do this and not others? What are the immunological tools that they use, and why is it not universal? So this gets us to a little bit of background theory, a little bit of conceptual development. Is more investment in defense inherently better? Should all of the lake populations invest heavily in defending against the tapeworm? Or is that not always a good idea? There are some people who will say that more and more investment in defense is always a better idea. But we don't want to talk about these people this week. So the alternative is that excessive investment in defense could be a liability instead of a benefit. And to give you a graphical version of this, consider a quantitative trait on the x-axis here, which is the magnitude of investment in immunity. And the more you invest in immunity, the lower your parasite load in blue, and the higher your fitness. Fewer parasites is good. But of course, at a certain point, you put so much into immunity that you have no infections. And so additional investment in immunity yields no additional benefit. When that's the case, you still accrue additional fitness costs. Even once you're completely immune, additional investment in immunity continues to add costs, even though it's not adding benefits. And so if we think about the net benefit, the difference between the benefit from killing parasites or the costs, we get a net benefit curve where there may well be an optimum. And all else being equal, you might think that a population would evolve some intermediate value of investment in immunity. So here's our phenotype distribution. They've optimized, and on average, they are at the optimum, not too much defense, but not too little. So one reason that populations might differ in their level of immunity is maybe that optimum is higher or lower in different lakes. So the ecological context, the parasite exposure rate, predation, food income, parasite genotype might shift the cost and benefit curves. Alternatively, it's worth noting that actually the population might not always land at the optimum. And the reason is this fitness landscape can be asymmetric. Yes, it's stabilizing selection, but if you go too far to the left, then you're susceptible, you get parasitized and die. If you go too far to the right, you survive, but you have maybe an incrementally worse cost. And when that's the case, the equilibrium for the phenotype mean of the population is actually a bet hedging strategy. It's like when you're on the top of a mountain, you don't stand right next to the cliff. You step away from the cliff a little bit. So think about the fitness cliff on one side, you get parasitized, makes you step downhill a little bit from the peak to get away from that steeper cliff. So we might expect some populations to evolve to stand away from the cliff and others to stand right up on the summit. So asymmetric selection can favor excessive defenses. The problem is we have relatively little data on costs of immunity in wild populations. How often do we really see immunity that's detrimental to the individuals that use it? And so this is where my postdoc Natalie Steinel comes in. So she was doing an experiment. Oops, I forgot I changed the picture there. She's now a professor at the University of Massachusetts Lowell. She was doing an experiment where she was injecting stickleback with artificial antigens to track their immune response. And although that experiment did not end up working very well, she came to me one day and said, Dan, I noticed something very strange. In some of the fish that I injected just with alum, which is a vaccine adjuvant, I see this strange phenotype. Have you seen this before? And so I walked across the hall to the dissection room and took a look. And what she saw is this. This is a normal stickleback. You see the liver, which is being moved around there, is very loose. It's not attached closely to the stomach. It's not attached to the body wall. But what she was seeing was this. The entire body cavity of the stickleback was glued together as if somebody sprayed glue all over the organs. The body wall was glued to the stomach. The stomach was glued to the liver, and you could barely separate them. Now, I had, in fact, seen this before. I had seen this regularly for 15 years. From the very first stickleback I dissected, I had seen some of this phenotype. But I always thought that maybe this is because I didn't freeze the fish quickly enough after I euthanized them. Or maybe I put them in formaldehyde and it didn't fix them internally quickly enough. And so I just dismissed it as strange, but maybe an experimental artifact. So I had seen something every day and yet not noticed it. And it took Natalie pointing it out to me in the laboratory for us to become interested in what turns out to be a very interesting phenomenon. So when we inject fish with saline solution, we have a normal histological section like you see in the middle here. This is a section of the gut. You see that red in the middle is the stomach wall. In the top right is the spleen. On the right side is another histological section where you see a lot more purple tissue, and the spleen is connected to the intestine with collagen and fibrinogen. This is a fibrosis response to this alum immune challenge. And that fibrosis response is basically scar tissue. And this scar formation is a very common problem in humans. And it turns out that it does not happen spontaneously in stickleback, at least when they're young, but it does happen when we expose them to tapeworms or when we inject them with alum. And so to drive that point home, here's the frequency of fibrosis in infected versus uninfected fish of two genotypes, F2 intercrosses and Roberts backcross fish. And you can see the frequency of fibrosis is much higher in the infected fish than the uninfected ones. I should note the uninfected fish all were fed tapeworms, but in many cases they successfully killed the tapeworms. So they're not presently infected, but they did see a tapeworm. When we have control fish where we never feed them the tapeworm, the fibrosis frequency is just 0, always in the laboratory. The gosling fish, on the other hand, the back cross to gosling, do not do fibrosis at any large frequency. So we have genetic variation here in the fish's fibrosis response to tapeworm infection. That matters because having fibrosis reduces tapeworm size dramatically. Again, this is a log scale on the y-axis, and I'm showing you the trends for the 3 different genotypes. F2 hybrids in the middle, back crosses to Roberts down below, the smaller worms, and gosling fish up on top. And moreover, this allows the fish sometimes to kill the tapeworm. So the tapeworm gets surrounded by a wall of connective tissue to form granuloma. And we frequently found these granulomas either with tapeworms or with dead tapeworms or with balls of cells that were just dissociated and no longer intact, but had tapeworm DNA. So we can confirm for certain that these granulomas lead to death of the tapeworm. And you need to have this fibrosis response in order to generate the granuloma. So this fibrosis, the scar tissue formation, is essential for surrounding the tapeworm when it's small and killing it. Now, I don't want you to think that fibrosis is the entire story. When we do a path analysis with our F2 hybrid and backcross fish, we see that Robert's ancestry affects cestode mass through multiple different genetic pathways. Directly, via body size, via fibrosis, and via reactive oxygen production. And all of these pathways contribute to reducing cestode mass. Fibrosis is the overall largest effect size so far. Now that's in the laboratory. Does this hold in the wild? So to check this, we collect fish in the wild from populations that have variation in fibrosis and reasonable numbers of tapeworms. And what we found is that the more severe the fibrosis in fish from a given lake, the smaller the tapeworms are. So individuals with more fibrosis have smaller worms. Across lakes, lakes where fibrosis is more common have smaller and fewer tapeworms. Lakes where fibrosis is very rare have very high infection rates. So at the level of individual fish and also among populations, fibrosis is associated with smaller, fewer tapeworms in the wild and in the laboratory. And yet we see lakes with fibrosis and we see lakes without fibrosis. Just yesterday afternoon, I dissected 100 stickleback from Merrill Lake on Vancouver Island. We had 80% infection rate and I saw 0 fish with fibrosis. And this lake is just a few kilometers away from the lake that has 0 tapeworms and 80% of the fish have fibrosis. Now, if you're paying attention, you'll say, wait, why do they have fibrosis if there's no tapeworms? And the answer is if they ingest a tapeworm and they kill it, they still get the fibrosis. And the fibrosis can last for at least 3 months after that experimental exposure. And so the fibrosis remains a very long time. And so what we're seeing is the ghost, the immunological ghost of a former infection attempt. So why doesn't everybody do this? And I think the answer is it's costly. In the laboratory, when we compare fish with or without a cestode, females are equally likely to be reproductively mature regardless of whether they have a cestode. But if they have fibrosis, they are much less likely to be reproductively mature. And this is statistically controlling for ancestry. In the wild, we see the same trend. This is from a single lake on Vancouver Island. Individual females with fibrosis are less likely to be reproductively mature. Individuals with tapeworms, there's no significant trend. So tapeworms themselves don't limit reproduction in these systems. Instead, tapeworms induce fibrosis that limits reproduction. Now, a caveat here, once the tapeworms get really, really big, this might no longer hold. So this might change, the answer here might change seasonally. We also test this with males. A postdoc, Stephen Delisle, collected nesting and non-nesting male stickleback from Vancouver Island from 2 lakes. And in both lakes, males with fibrosis were less likely to be nesting with eggs or fry than males that had fibrosis. So fibrosis is bad for female reproduction. fibrosis is bad for male reproduction. And we've confirmed this in multiple lakes. So here we have this pyrrhic victory. In a few lakes, fish are able to effectively limit tapeworm infection, in some cases almost completely, but they end up with this very severe immune pathology that prevents them from reproducing successfully. And they may have won the battle and yet reduced their fitness dramatically in the process. So coming back to this notion of costs and benefits, what I would argue is that in Roberts Lake, they induce fibrosis that reduces cestoid growth, but it really gets rid of their fecundity. Stickleback from Roberts Lake, on average, generate 20 eggs when they're reproductively mature. In contrast, stickleback from Gosling Lake don't do fibrosis. And even when they have a tapeworm, they can generate as many as 200 eggs in a single female. 10 times more eggs, but they get a big tapeworm. Sounds like a good deal. So the question is, it seems like we have this axis of variation from a tolerance strategy that accepts the infection to a resistance strategy that accepts low fecundity. And the question is, why do they have different optima? And the answer is, I don't yet know the ecology here. I can't tell you why the optimum for one population might be different from the optimum for another. Could be any of a wide variety of reasons. But what I'm going to shift to now for the very end of my talk is let's look into the genetics of this system. This is exciting because this is a naturally evolved model system for fibrotic disease. Humans get fibrotic disease. Fibrosis is a major contributor to mortality in cancer cases, pulmonary fibrosis, cirrhosis of the liver, renal kidney failure. And so here we have an opportunity to study the genetic basis of a naturally evolved immune response generating severe fibrosis. In short, the Roberts Lake fish seem to have gone for a very, very strong type 2 immune response that generates severe inflammation and severe fibrosis. And the Gosling fish are avoiding that strategy but accepting infection. So we begin with some genetics, just confirming that there are differences in response. So remember, in the wild, marine fish don't get infected naturally. And neither marine fish nor Gosling Lake fish exhibit fibrosis in the wild. Both Roselle and Roberts Lake fish do. When we inject any of these fish laboratory raised with saline solution, there's no fibrosis. When we inject them with alum, they all get fibrosis. Although some of them get it a little faster and some of them clear it a little faster, they all have the cellular genetic machinery to do fibrosis when they choose to do it. But if we inject the fish with tapeworm protein, then only the Roselle and Roberts Lake fish that normally exhibit fibrosis proceed with a fibrosis response in the laboratory. So if we think about the cascade of events that happen during an immune response, you recognize that you have an infection, you signal to do something, you do something, you attack it, and then you damage or kill the parasite. Clearly, all of the populations share these last two steps. They are all capable of doing fibrosis. So the genetic variation is earlier in this cascade. Then we did transcriptomics of Roberts and Gosling fish, and what we find is that they actually show very similar transcriptome responses to the presence of a tapeworm. There are only 10 genes that show genotype by infection interactions, compared to 54 genes that are up or down regulated in both populations when they're infected, which suggests to us that the initial step of recognition is probably similar in both populations. So it's not recognition and it's not ability. Instead, there's a switch somewhere along the way that makes the difference between turning on or turning off fibrosis. So I don't expect this to be an MHC gene or a pattern recognition gene that detects a parasite. So to figure out what the genetic basis of this is, we combined QTL mapping with transcriptomics with population genomics to try and find the intersection of these different methods of mapping to find loci that, for example, are located where we can map a QTL for phenotype and show a signature of selection. So we've mapped infection rate, cestode mass, fibrosis, reactive oxygen by experimentally infecting 740 hybrid stickleback with cestodes. So we see, for example, a very clear QTL for cestode mass on linkage group 12. We see a QTL on linkage group 2 and 8 for fibrosis. We see reactive oxygen on linkage group 15. Cestode mass is also on linkage group 15. Some of these QTL locate to the same place. So there's some chromosomes that contribute to fibrosis and cestode mass. Some that contribute to reactive oxygen and cestode mass. We then complement this with population genomics. We did pool-seq, whole genome sequencing on 100 marine fish as an outgroup, 100 Roberts Lake fish, and 100 Gosling Lake fish. And for the whole genome, we can build a phylogenetic tree with branch lengths that tell us how related these populations are. And then we scan through the genome to find loci that show accelerated evolution along one branch. So this is the genome-wide average that I'm showing you here. But at a particular locus, we might, for example, see 2,000 or 3,000 times longer branch length in Gosling Lake, whereas Roberts and marine fish are basically identical. This suggests accelerated evolution, natural selection in Gosling Lake. Now, it turns out that this is the common pattern. Almost all of the targets of selection in our QTL were selection acting in Gosling Lake, which is surprising because Roberts Lake is the lake with the derived fibrosis phenotype. Roberts Lake is where they have gained the fibrosis ability. So why is this selection happening in Gosling Lake? And I'll show you this in just a second. So here's an example of the data. This is inside the QTL for fibrosis on linkage group 2. And we have an area in the red box with high allele frequency difference, with a high population branch statistic specifically in Gosling Lake. So the allele frequency difference just tells us that Roberts and Gosling Lakes are different. The population branch statistics tells us where the evolution happened, in which population, and it's very much in Gosling Lake. Inside this area of selection are just 5 or 6 genes. One of those is PU1, which a Nature paper a year and a half ago identified as controlling fibroblast polarization to generate tissue fibrosis. So this is a fantastic candidate gene. We now have knockouts with CRISPR that we're growing up in the laboratory to test. So in a year, we should know for certain if PU1 is regulating fibrosis in stickleback. Now let's look at cestode mass. We have a QTL on linkage group 12. Inside this, we see a strong target of selection that includes a gene STAT6. And this is exciting because in Gosling Lake and a couple of other lakes with big happy tapeworms, we see a frame shift deletion in one of the early exons of this gene that disrupts the gene function. Marine fish and Roberts Lake fish have intact STAT6. Gosling Lake, this red window here, involves a predominant deletion. That's important because in laboratory mice, STAT6 is a very well-known regulator of susceptibility to helminth infection. STAT6 knockout mice in the laboratory get nematodes much more readily than the wild type mice do. And so again, we have a very promising candidate gene. We know STAT6 regulates helminth immunity. And here we see that in stickleback, a STAT6 deletion is under positive selection to fix this deletion in a population that has, in essence, chosen to accept a tapeworm infection. This result is only a couple weeks old, so we're very, very excited about it. So now we're delving into understanding more about what STAT6 does and looking at the downstream gene expression pathways. Just real quickly, we do see some other candidates also in the same QTL on linkage group 12. We see a big deletion, a whole gene basically, a 3,000 base pair deletion in a gene that's involved in oxidative reaction of xenobiotics. So here's the puzzle now. And I'm going to end with a brief hypothesis. Marine stickleback colonized lakes, and all of these fish were exposed to tapeworms that are abundant in fresh water. So they have high exposure. Both lake populations have lower infection success than marine fish do, so they gained resistance. They both gained resistance. So there was some gain of function in both. And yet, the cestodes grow well in both marine and Gosling lake fish. Fibrosis is only in Roberts Lake fish. So we would think that natural selection was doing something special to cause fibrosis in Roberts Lake. So we should see selection in Roberts Lake for fibrosis, but that's not true. We see the selection in Gosling Lake. So why is that? My speculation here is that Gosling Lake might have evolved fibrosis early on, but then abandoned it and suppressed it. And here's why I think that's true. When we do our gene expression analysis, we find differential expression of genes involved in fibrosis, both between infected and uninfected fish and between fibrotic and non-fibrotic fish. And one of the main genes that's differentially expressed is HNF4 alpha, which is an upstream regulator that suppresses fibrosis. So more HNF alpha means less fibrosis. Roberts Lake fish, when they get infected, their HNF4 alpha does not change. It goes up just a tiny bit. Sorry, it actually goes down a tiny bit. Gosling Lake fish, when they get infected, their HNF4 alpha goes through the roof. So Gosling Lake fish are turning up a fibrosis suppression system. So they are actively getting rid of fibrosis. So here's my idea. Both populations evolved fibrosis 10,000 years ago. Gosling Lake fish decided resistance is futile. And so they increase HNF4 alpha to suppress fibrosis, and they knock out two different genes that contribute to fibrosis. They knock out PU1 and they knock out STAT6. All three of these changes involve natural selection in Gosling Lake, not Roberts. So selection is favoring genomic changes that are suppressing fibrosis rather than favoring the genomic changes that accelerate fibrosis to begin with. So almost done here. We are working on doing the gene manipulations to knock out these genes experimentally, and I am hiring a postdoc to do this work. We've already started some of it, but my postdoc Foen Peng, who's working on this, is leaving for a faculty job. So if anybody's interested in doing CRISPR-Cas9 editing of stickleback immune genes, please get in touch. We also now need to do QTL mapping, not of Roberts versus Gosling, but we need to cross them both back to the marine fish. And what we might find, I predict, is that we find fibrosis genes in both populations, even though the Gosling fish don't do fibrosis today. And we now have single cell RNA-seq data because of my former postdoc, Lauren Foos. This is single cell RNA-seq data from head kidneys and stickleback. And we now know that Roberts Lake fish have very different immune cells than Gosling and marine fish do. So we know there are population differences in which cells are present and differences in what genes the different kinds of cells express. And lastly, a former postdoc, Milan Vrtlek, has done these vaccinations with tapeworm protein and alum into a wide diversity of fishes. And we know that there's phylogenetic signal in the intensity of fibrosis response to alum and to tapeworms across the diversity of fishes. Almost no fish except stickleback and carp respond to Schistocephalus solidus tapeworm protein. But most fish respond to alum, but to different degrees. And so we're very interested now in the genetic diversity across the entirety of fishes in this fibrosis response. Now, all of this is material that I missed for 10 years as I ignored the tapeworm and I ignored the fibrosis. And so if there's any lesson that I want you to learn, it's that when you see something and think, that's strange, I wonder what that is, it's probably worth looking into. Because in the process of delving into the tapeworm and the fibrosis that's associated with it, we now know that ecology and immunity jointly regulate cestoid prevalence across lakes. And we know that some populations have adopted this pyrrhic victory of a costly fibrosis, and other populations seem to be actively evolving to suppress that fibrosis and accept tapeworm infection in exchange for reproduction. And with that, I need to thank a wide variety of institutes that funded this work and many, many people who contributed to the work, and I will be happy to take any questions.
We're all muted, but we're clapping. Thank you very much.
Thank you.
Thank you. Do you think we should leave your slide or remove it so we see faces of people for the question period?
I can remove the slide and then.
Okay. So the floor is open for questions. I'm gonna let people unmute because I know it takes a while. Do I see raised hands? Because I can start with the question so people can. Okay, so yeah, everything is great. I'm like, yes, that's cool. One of the great things about the stickleback schistocephalus system is that it's everywhere in the Northern Hemisphere. We have it in Japan, in Russia, in Europe, in Canada, East Coast, West Coast. Does it mean that we could go and sequence STAT6 or the other, or PU1, and look at the allele and predict fibrosis response or the or just tolerance?
I hope so. So, so STAT6. 2 weeks ago, my research group had our weekly lab meeting, and we were reading a paper by Andrea Graham and her group, where they took STAT6 knockout mice. This is one of my favorite papers. I love this paper. Andrea's group took STAT6 laboratory mice and put them in big cages in a field in New Jersey, in the wild, and exposed them to tapeworms or not. Not tapeworms, nematodes. And she found that STAT6 knockout mice, which are very well known in the laboratory to be susceptible to worms, in the field, she did not find the same phenotypes. And all of the immune cell population and immune responses that are very well characterized in the lab didn't exist in the wild. So the 5,000 published articles about STAT6 knockout mice might only be relevant to the laboratory, okay, which is a beautiful result for an eco-immunologist, right? Mm-hmm. So we were reading this paper and talking about STAT6, and my postdoc, Foen Peng, said, let me check and see if STAT6 is one of the genes in our QTL. And sure enough, it's the strongest target of selection in that QTL. And then he looked at the sequence and he said, oh, there's a big deletion. Well, not a big, 8 or 10 base pair deletion. That's a frame shift inside Gosling Lake and one other lake, but not Roberts. So literally our attention was only drawn to STAT6 2 weeks ago. Mm-hmm. So our very first order of business is I have 100 populations worth of DNA in the freezer. We need to go and sequence STAT6 from all across Vancouver Island and Alaska and correlate it with tapeworm size and abundance and our more limited fibrosis data. I'd love to do the same for the Northeast and Iceland and Europe and Japan. We just don't have, I don't have the DNA samples, mm-hmm. But very high priority for us is just sequencing STAT6 from all around the world as best we can. But I especially want to do that when we know that there's fibrosis. So we know there's fibrosis in some lakes on Vancouver Island and not others. We know there's fibrosis in some lakes in Alaska, but not others. Greenland, Lake Matthews dissected large numbers of fish from about 100 populations a year ago. He says he saw no fibrosis at all. So I'm not sure I would bother too much in Greenland, but I mean, maybe it's worth trying. So I think first I sort of want to just get as much data on fibrosis and tapeworm size and abundance as I can.
Mm-hmm. Okay. there any questions? Xavier?
Hi, thank you for this amazing presentation, Daniel. I was wondering, are you hearing me?
Yeah.
Is it working? Okay. So even on Vancouver Island, do you expect that all the population that does not react to cestodes have the same mechanism for this suppression? Do you think like that STAT6 will be interesting in all those populations, or do you expect actually like multiple pathways?
That's a very good question. In general, I'm a firm believer that evolution is usually not very parallel. And I say that as a stickleback biologist, where the armor plating, Ida in stickleback, is maybe the best example of parallel evolution that we have. But in general, I think that's exceptional and unusual. So we do have, I didn't have time to talk about, my talk was too long anyway, but we do have population genomic data from about 70 lakes on Vancouver Island with parasite frequency, and about half of them we have tapeworm size as well. And so we've done genome-wide association mapping, GWAS, across populations on Vancouver Island. And the advantage of GWAS is it will not detect genes that are uniquely adaptive in one place but not other places. You're really looking only for the genes that are associated with parasite size or abundance or fibrosis very generally. And we see lots and lots of genes correlate, lots of genetic markers with this RAD-seq data that are correlated with tapeworm size across these lakes, or correlated with tapeworm number across these lakes. And many of those SNPs are very close to genes for fibrosis or reactive oxygen, which suggests to me that there's some amount of parallelism, but it's not, there's probably no universal single solution.
Yeah, thank you. Thank you for the details.
Other questions? Okay, well, okay, Johan?
Yeah, I have a quick question. So I think it's, well, thank you for the nice presentation, first off. And it seems pretty clear that, like you say, Robert's fish have this strong immune response and everything. But that doesn't really explain why you don't find any tapeworm in the fish when you sample from the nature, right? Because they get infected by the tapeworm at 15 or 20%, similar to the gosling. So why don't you find the tapeworm?
That's a lingering problem for me. So here's my explanation with the caveat that I might be proved wrong. So remember, the Roberts Lake fish, when they do fibrosis, they often encapsulate the tapeworm in a granuloma and they kill it. And then there's no tapeworm left. And then the fibrosis persists after that. The Roberts Lake fish in the wild frequently have fibrosis. When we take them into the laboratory, they don't develop fibrosis spontaneously. So that fibrosis is being induced by something. And the only thing that we know of in nature that induces it is schistocephalus, unless somebody's going around injecting vaccine adjuvant into these fish in the wild, which is not likely. So I think they are getting the tapeworms, they are killing them quickly enough that we're not seeing the tapeworms, but we do see the echo of that, the scar tissue that remains. To prove that, what I need to do is get environmental DNA data to show that the tapeworms are present in the copepods in the lake. Get stomach content data and do eDNA on the stomach contents to prove that they are ingesting the copepod, that they're ingesting the copepods with tapeworms. And then that would be, I think, the convincing evidence. we've tried that and we've gotten no result, but we also get no result when we do the environmental DNA in Gosling Lake, where we know there are lots of tapeworms. So it's a but then when we do the eDNA with laboratory infected copepods, we get positive results. So we know our protocol works. So my guess is that when we're doing our eDNA, we're doing it when we do field work in May and June. Maybe I need to be doing it in November. Maybe I need to be doing it in March. Maybe I need to be, I don't know the right time of year. And it's also possible that plankton toes might not be the right way to catch schistocephalus infected copepods. Maybe the schistocephalus infected copepods stay right near the benthos, or maybe they move to a certain part of the lake or a different depth than we're sampling at. So we're clearly missing the tapeworms because they do exist in Gosling Lake and we don't see them with eDNA. So something's wrong. And it's something about our sampling.
Yeah, so wasn't there a seasonality to this parasite, that they get infected at a certain time? Or how long does it take for the Robert's fish to kill the worm in your experiment in the lab?
Days.
Days. Okay, so you have a really short window.
Yeah, days to weeks. The seasonality is not well characterized. It's one drawback of doing field work on Vancouver Island and living in Texas and then Connecticut, is I just don't have year-round access to these lakes. And I'd love to find somebody who lives there. And if I get some extra grant money sometime, I'll probably just hire somebody at Bamfield Marine Science Center to visit a couple of the lakes twice a month and sample. But I think the answer might be no, because I would have guessed yes, but at this point I do have fish samples from multiple times of year, and we see tapeworms throughout the year. So I just yesterday was dissecting fish from Vancouver Island that were collected in mid-October for us, and they're full of tapeworms. And these are young of year juveniles, so they've been infected in the first few months of their life.
Okay, thank you.
Other questions?
I have a question.
Yeah.
So do you think that fibrosis is automatically too costly, or could some kind of an equilibrium could evolve in some populations, maybe between having a fibrotic response, so you maybe get an infection sometime, but some other time you don't, and the cost on the reproductive cost is not so heavy?
It's possible, but so far the reproductive cost of fibrosis is quite repeatable in many lakes. So we don't see any evidence that that's separated. What we're trying to do next, one of the paths that we're working on is to experimentally try and determine the mechanism of the cost. So because we can inject fish with alum and cause fibrosis, we can do that and measure sex hormone production. So I have an undergraduate who's asking, does this impact sex hormones? And then we injected fish with alum or saline, and we're tracking gonad development. Does it physically, my first guess is that it physically constrains ovary development in females. Females need big ovaries to be fecund, and the scar tissue really just glues together the body, which I think makes it harder to expand. There's just it's like putting chains around their ovaries. That led me to expect that the cost of reproduction would only be in females but not males, which isn't true. So we know that males also have a cost. So we did an experiment, or I should say Ken Thompson at UBC did an experiment this spring, injecting fish with saline or alum in the ponds at UBC campus, and then actually measuring their reproductive success by parentage analysis of the offspring. So we're trying to confirm it that way. And then we're working with George Lauder and one of his students, Dave Matthews at Harvard, who are doing biomechanics to look at swimming kinematics of fish with and without fibrosis. and we're working with somebody doing the metabolics and the metabolic cost of generating fibrosis. So we're really trying to get all angles. Is it energetics? Is it physical constraint? Is it swimming performance? But it's clear that there is a pretty big cost, that it's the fibrosis per se, because when we induce fibrosis with a vaccination, they're less fecund. We can also use that to experimentally confirm that it's the fibrosis that suppresses tapeworm growth. We can take gosling fish, we give them tapeworms. Normally those tapeworms would grow and be happy, but we can inject those fish with alum, force them to do fibrosis, and ask whether the tapeworms don't grow as well in fish that would normally permit growth. And then in Roberts Lake fish, we can treat them with a drug that we're testing out this fall that is supposed to reduce, it's a drug to treat fibrotic disease in humans. So it's an anti-fibrotic drug. So if we give that to fish, can we prevent fibrosis and rescue tapeworm growth? So if we can do all of that, we could prove that if you force fibrosis, you get small worms. If you use drugs to stop fibrosis, the worms get rescued. Then we'll really know absolutely that this is the cause. And then the gene knockdown with CRISPR-Cas9 will confirm the gene involved.
Thank you.
So it's a 1:45. So it means that there was a lot of interest with questions. Thank you very much, everyone, for attending, and for the grad students and the postdocs who are staying after, or professors, if you want, to talk with Daniel. We'll give him a 5-minute break. Huh? Let's give him 5 minutes, and then we're still on the Zoom, and we can ask other questions and discuss in general with him. Okay, thanks again.
Thank you all.